Learning outcomes. Dear students, after studying this module, you shall be able to know about what is chromatography, the various types of chromatography and planar and about paper chromatography. Now students, let us start with the introduction of this module. Chromatography is a group of technique in which a mixture is separated into individual components due to differential distribution of the solutes between a stationary phase and the mobile phase. Chromatographic techniques are invaluable tools of analysis as the components of mixture are first separated, facilitating the analysis of complex mixtures. The history of chromatography dates back to early 1900s when a Russian scientist, Michael Sweat, successfully separated a mixture of plant pigments using calcium carbonate and alumina packed in a glass column. In his experiment, he made a solvent extract of homogenized plant leaves and applied this extract onto the calcium carbonate column. The sample was allowed to travel through the column along with the solvent under the gravity. The components of plant pigment mixture separated into different colored bands in the column. Sweat coined the name chromatography from Greek word chroma meaning color and graph meaning writing. Two British researchers, Martin and Sinch, years later amended upon Sweat's chromatographic technique and devised a technique known as paper chromatography. They were able to use this technique to separate the amino acids in protein hydrolysate and were awarded the Nobel Prize in 1952 for their work. Over the last century, the technique of chromatography has provided significant contribution in the field of molecular characterization in addition to the purification process. Quantitative chromatographic analysis is invaluable in pharmaceutical industry for research as well as for quality control. Similarly, biotechnology industries widely use chromatographic techniques and in many cases alternative methods are not available. As an example, chromatographic techniques have been applied for separation of stereoisomers that are very similar in structure and properties. The development of biotherapeutics would have been impossible without chromatography based purification strategies. The reasons for popularity of these techniques lie in their unique sensitivity, flexibility and scalability. Moreover, chromatographic separations are relatively fast and simple and afford considerable ease of operation compared with the other instrumental techniques. Chromatographic separation involves a stationary and a mobile phase. The analyte mixture interacts with the stationary phase. Depending upon different types of interactive forces of components of the mixture with the stationary and the mobile phases, some molecules are detained in the column more than the others. The difference in the migration rate of the components of the mixture brings about the separation as the mobile phase moves and flows out of the system. The mobile phase can be either liquid or gas. The stationary phase can be solid or liquid. Next is about the chromatogram. A chromatogram is the visual output of a chromatograph. Different patterns or peaks are obtained on the chromatogram during an optimal separation wherein the different peaks correspond to different components present in the separated mixture. Next, we will be studying about types of chromatography. Chromatography can be broadly divided into two types. One is the planar chromatography and the second one is the column chromatography. So, students, let us start first with planar chromatography. Planar chromatography, in this type, the stationary phase is supported on the fibers of a paper or on a flat plate, whereas the mobile phase flows or moves by capillary action or under the influence of gravity through the stationary phase. The two major types of planar chromatography in use are paper chromatography and thin layer chromatography, also known as TLC. Next is column chromatography. The stationary phase in case of column chromatography is held within a narrow tube via which the mobile phase moves through either by gravity or by pressure. Column chromatography can be further differentiated based on the types of stationary and mobile phases and the kinds of equilibria involved in solute transfers between the two phases. There are several classes of column chromatography such as gas chromatography, high performance liquid chromatography also known as HPLC, etc. The column chromatography types can be further classified, and this classification has been explained in this very table, wherein in one column we have the general classification, the other column shows the specific method name, the third column shows the stationary phase, and the fourth column shows the type of equilibrium. Let us first start with liquid chromatography, where the mobile phase is liquid. There are specific methods under this type. First is liquid, liquid or partition, where the stationary phase is liquid adsorbed on a solid, and the type of equilibrium is partition between immiscible liquids. Next is liquid bonded phase, where the stationary phase is organic species bonded onto a solid surface, and the type of equilibrium attained is partitioning between liquid and a bonded surface. The third type is liquid solid or adsorption, where the stationary phase is a solid and the type of equilibrium attained is adsorption. Next is ion exchange, wherein the stationary phase is an ion exchange resin and the type of equilibrium is attained by ion exchange. Next is size exclusion, wherein the stationary phase is liquid in interstices of a polymeric solid and the type of equilibrium attained is by partition or sieving. The second type is the gas chromatography GC, wherein the mobile phase is gas. Herein the types included are gas liquid, wherein the stationary phase is liquid adsorbed on a solid and the type of equilibrium is partition between gas and liquid. Next is gas bonded phase, where the stationary phase is organic species bonded to a solid surface and the type of equilibria is partitioning between liquid and a bonded surface. Next is gas solid, where the stationary phase is solid and the type of equilibrium attained is by the process of adsorption. Now, we will be moving on to more detailed study of a planar chromatography. In all of the different forms of chromatography, a rapid dynamic equilibrium of molecules exists between two phases, that is the stationary phase and the mobile phase. For example, consider a mixture of molecules A and B dissolved in a mobile phase and flowing over a stationary phase. The molecules of compound A and B exist either as completely dissolved in the mobile phase or may be bound to the stationary phase. The equilibrium of the molecules between the mobile and stationary phases depends upon polarity and size of the molecule, polarity of the stationary phase and polarity of the solvent. Depending upon the three factors, the components of mixtures partition differently between the mobile and the stationary phase. For example, consider separation of a mixture containing compound A and B. Compound B has a greater affinity towards the stationary phase as compared to the compound A. When the mixture flows over the stationary phase, the molecules of compound A spend more time in mobile phase and move farther in a given amount of time. Whereas, since compound B has greater affinity towards the stationary phase, the molecules of compound B move at a slower pace through the stationary phase particles. As a result of this, the components of the mixture can be gradually separated. A suitable solvent mixture can be selected as a mobile phase to achieve the desired separation. Next, we will be studying about paper chromatography. In paper chromatography, substances are distributed between the paper as a stationary phase and a suitable mobile phase. The chromatography paper has highly uniform texture and thickness. It is made from cotton cellulose and has low content of additives. For the separation of the mixture, a discrete spot is applied on the paper. The components of the mixture partition between two phases. Aqueous phase consisting of water adsorbed in the pores of the chromatography paper, also known as the stationary phase, and solvent, that is the mobile phase, that moves over the paper due to capillary action. The compounds in the mixture get separated due to differences in their affinity towards the two phases. An additional factor could be adsorption of the components of the mixture by the filter paper. Paper chromatography is a useful technique for separation and analysis of molecules because it is quick and requires small quantities of material. The equipment for paper chromatography consists of a vapor-tight glass chamber with inlets for addition of solvent in addition to supports for solvent troughs and chromatography paper. In paper chromatography, a volatile solvent is used to carry the components of the sample along with the paper. This is called as developing solvent. The choice of solvent depends on the chemical nature of the material to be analyzed and needs to be optimized for every type of mixture. Usually solvents such as ethanol, methanol, isopropanol, acetone, diethyl ether, chloroform, methylene chloride, formamide, acetic acid, ammonia, cyclohexane, benzene can be used alone or as a mixture as a mobile phase. The general procedure that is followed in case of paper chromatography follows the sequential steps, wherein the first step is preparation of chromatographic chamber. The procedure for paper chromatography starts with equilibration of chamber with solvent vapors. The pre-saturation of chromatography chamber is carried out well in advance before the development. The saturation of the chamber with the solvent may be facilitated by lining the chamber with filter papers wetted with the developing solvent. The next step under this is preparation of paper. The paper may be used such as where water adsorbed in the fibers acts as a stationary phase. Alternatively, paper may be impregnated with a polar solvent other than water such as buffer, salt solution, methanol or glycol. Paper may also be modified with silica, diatomaceous earth and alumina. Such type of modification papers would illustrate adsorbing capacity of the respective modifiers. Paper may be made hydrophobic by impregnating with silicon or other non-polar solvents. Paper may also be acetylated so that it becomes more hydrophobic and retains a similar hydrophobic component. The third step is application of samples. The mixture to be analyzed is dissolved in a suitable solvent. A fine line is drawn on the filter paper so that it is few centimeters away from the level of developing solvent. The dissolved samples are applied on the spots diameter of about 6 to 10 millimeter that lie at least 3 centimeters apart on the line drawn on the chromatographic paper. The sample application is usually performed using micropipettes. The sample application may be repeated after solvent evaporates from the previous spots to concentrate the sample. The next step is the development of the chromatogram. After preparing the chamber and spotting the samples, the paper is ready for development. Depending upon the type of development, paper chromatography can be classified in several forms such as ascending paper chromatography, descending paper chromatography, ascending descending paper chromatography, radial chromatography and two-dimensional paper chromatography. Now, let us take one by one each of these. First is ascending paper chromatography. Here the developing solvent is poured at the bottom of the chromatographic chamber. The solvent travels in upward direction due to capillary action. Next is descending paper chromatography. Here the solvent is allowed to travel down the paper. The mobile phase is kept in a trough located at the top of the chromatographic chamber and it is allowed to descend over the chromatographic paper. Next is ascending descending paper chromatography. It is the hybrid of both the ascending paper chromatography as well as the descending paper chromatography. The upper portion of ascending chromatography sheet is folded on a rod while allowing the descent of paper after crossing the rod. Next is radial paper chromatography. It is also called as circular chromatography. Here a circular filter paper is taken and the sample is applied at the center of the paper. After drying the spot on the filter paper, it is tied horizontally on a shallow chamber containing the solvent. A wick of the paper is dipped inside the solvent. The solvent rises through the wick and the composition or components gets separated in form of concentric circular zone. Next is two-dimensional paper chromatography. In this very technique, a square or rectangular paper is used. Here the sample is applied at one corner and development is performed at right angle to the direction of first run using a different solvent. This facilitates better separation of the components of the mixture. The next step is analysis of result. After the development, any strongly retained compound can be visualized as colored spots on chromatogram. Colorless molecules can be visualized by heating the paper in a hot air oven so that the substance in the spots char and can be seen as black spots. The paper may be sprayed with a solution of sulfuric acid before heating for better charring. Fluorescent materials can be visualized with ultraviolet light. Reagents specific for certain components may be sprayed on to make spots colored. Radioactive spots can be located with a detector or the X-ray film may be exposed to chromatogram. A few examples of paper chromatography application and their analysis has been shown in this table, where in one column we have the analyte component, the next column shows the solvent and the third column is for visualization reagent. First, starting with amino acid mixture, wherein the different solvents which can be used are as follows, that is N-propanol in addition with ammonia, phenol in addition with a buffer, N-butanol in addition with a buffer and acetone in addition with the buffer. And the visualization reagent that can be used for this purpose is ninhydrin. Next is the chlorophyll pigments. The various solvents used for the separation of chlorophyll pigments are isopropyl alcohol, petroleum ether and acetone. And the visualization reagent will be the own colorful spots. Next are sugars. The solvents required for this particular category are butanol in addition with ethanol and water, butanol in addition with acetic acid and water, phenol in addition with water. The visualization reagents that can be used for this class is ammoniacal silver nitrate, benzene 1,3-diol in addition with HCl and acetone. Next are food dyes. The solvents that can be used for this class are 0.1% NaCl solution and the visualization reagent will be the colorful spots. Next, lipids. Solvent used dichloromethane or methanol and visualization reagent. For fatty acid, it is 1% osmic acid which gives brown to black color. For cholesterol, it is acetic acid or concentrated sulfuric acid which gives pink color. Next, for analytes such as salt mixtures of sodium, magnesium and potassium, the solvents used are methanol and the visualization reagents used are dichlorofluorescein solution and silver nitrate solution. The distance travelled by any compound depends upon the partition of the compound between mobile and stationary phase. Therefore, the ratio of distance travelled by the compound as compared to the solvent, known as retention factor or RF, can be used to identify the components of the mixture. However, since RF value often depends on the temperature, solvent and the type of paper used in the experiment, the most effective way to identify a compound is to spot the standard substances next to unknown substances on the same chromatogram. Sample spots can be identified if they have the same RF values to known standard spots. If RF value of a solution is 0, the solute remains in the stationary phase and thus it is immobile. If RF value is equal to 1, then the solute has no affinity for the stationary phase and travels with the solvent front only. Next, we will be studying about the applications of paper chromatography. Paper chromatography has been used for separation of mixtures having polar and non-polar compounds such as amino acids, hormones, drugs and other biochemicals. It has also been used for the evaluation of inorganic compounds. Although paper chromatography has become very popular due to its simplicity, it suffers from various limitations such as long development time, poor separation, low accuracy in quantitative determination and low reproducibility. Now students, let us summarize all that we have studied in this module. Chromatography is a technique in which a chemical mixture is separated into individual components due to differential distribution of the solutes between a stationary phase and the mobile phase. The mobile phase can be either a liquid or gas. The stationary phase can be solid or liquid. Chromatography can be broadly divided into two types. One is planar chromatography and the other one is column chromatography. In all different types of chromatography, a rapid dynamic equilibrium of molecules exists between the two phases, that is the stationary phase and the mobile phase. The distance travelled by any compound depends upon the partition of compound between mobile and the stationary phase. Thank you.